Hello there, my name is Stanislaw Koper and I'll be presenting my presentation on early warning detection systems for bioterrorism and pandemic monitoring. It has been proposed that the next acts of terror may no longer utilize conventional weapons, and the preferred choice of biological and chemical warfare is becoming an increasingly tangible threat. Viruses in particular are invisible to the naked eye, thus anyone who is infected may not know that they are. So this causes for an extreme concern in the biodetection community. As seen with the SARS-CoV-2 pandemic, we were not prepared for anything of this magnitude. So in order to prevent these kind of situations happening, we require an integrated biodetection system for all types of viruses and other pathogens. In order to capture your bioaerosol of interest, there are many different types of techniques that can be utilized. Each one has its own pros and cons, and thus it's important to know the properties of your bioaerosol, as each technique could affect it positively or negatively. Furthermore, there are many types of downstream processing techniques that can be used to identify and test for positive results. In the diagram below, you can see all the factors from start to finishes that could have a significant impact in the final readings of biological aerosols. It is very important that each one of these factors are carefully considered when designing your experiments. Otherwise, potential errors could arise, leading to incorrect identification. Recently, a new type of fully automated sampler has been developed, which utilizes Digital Micro-fluidics, which have an inherent advantage of manipulating very discrete microliter scale droplets, and also able to conduct multiple experiments at the same time, with real-time results within 10 to 6 minutes. This detection system utilizes ELISAs, which are highly specific antibodies that only bind to certain antigens of desire, thus making them a very attractive choice compared to PCR in environmental settings can become contaminated and often not work. In order to understand indoor sampling, often computational models are conducted beforehand to understand how each variable may impact the sampling process, such as the positioning of the aerosol sampler, and many other variables that may include inactivation, biodecay. The biodetection community has a significant hurdle to overcome. We still require a lot of standardization and collaboration across all fields in order to create a successful detection system, as seen in many of the papers published.